Hello, my name is Guilherme. I am a PhD student in genetics and plant breeding at the Statistical Genetics Laboratory at Exalc USP. This work was done by me and other researchers from Exalc and Embrapa beef cattle. In this video, I'll talk about the trajectory of Megatrichus maximus in the last half century, especially about cultivar Miyagi. Megatrichus maximus, also known as Panicum maximum, is a key forage crop in Brazil, widely adopted in cattle and sheep farming. Our journey begins in 1959, when the 9th International Botanical Congress took place in Montreal, attracting researchers from all over the world. In this meeting, researchers discussed the taxonomy and classification of grass species. Up until this point, much of the taxonomy was solely based on morphological features. Scientists advocated for the use of cryptic features for the classification of grass species, such as anatomy, physiology, histology, cytology, embryology, and biogeography. In the 1960s, the French Institute IRD began its research with Megatrichus maximus as a model species to study apomixis and its inheritance. Researchers from IRD found that the center of origin of Megatrichus maximus was in East Africa. Thus, expeditions were made to Tanzania and Kenya in order to collect genetic material to compose the germplasm bank. In 1982, IRD signed a collaboration agreement with Brazil's Embrapa. On the occasion, 426 accessions of the germplasm bank were transferred and introduced into Brazil for research and breeding purposes. Cultivar Tanzania was launched in 1990, Mombasa in 1993, and Masai in 2001, directly through the exploration of the variability already present in this germplasm. In the early 2000s, a farmer from the Brazilian state of Mato Grosso identified a mutant plant in a Mombasa field and soon shared its seeds with neighboring farmers. This plant had compact inflorescence and reduced seed sharing, valuable traits in agriculture. These characteristics are commonly selected during domestication of wild crops, and appear in grasses crucial in agriculture, such as rice, wheat, and maize. Some researchers believe that Miyagi seeds were shared among farmers from different countries and that the cultivar Moon River, launched in Thailand in 2019, is actually the same as Miyagi. Genetic studies are still needed to confirm this hypothesis. The studied dataset is comprised of one sample of cultivar Zuri, one sample of Milênio, one sample of Paredão, two samples of Mombasa, 6 samples of Miyagi, and 6 samples of a sixth genotype present in the germplasm bank called Gen01. The samples were subjected to genotyping by sequencing. The reads were aligned using the Megatrichus maximus reference genome released by CIMMYT from CGIAR. SNPs were called using the GATK software and stored as VCF files. Genotyping was done using the UpDog-R package and considering the norm model. After genotyping, the genetic relationship matrix was calculated using the AGAge matrix package and the Van Raden method. This matrix was used for hierarchical clustering and other subsequent analysis. This is the resulting G matrix. The phylogenetic tree separated samples from different cultivars and clustered samples from the same cultivar. Samples from Miyagi and Mombasa were clustered together in the same group. This principal components analysis is the result of the composition of the G matrix. There is one cluster containing all the samples of Gen01. There is also a cluster containing samples of Miyagi and Mombasa with no clear separation. In the first 3 components, we observe mainly differences among cultivars. By rotating the space created by the principal components, we can better see the relationships between the samples. We can also explore other dimensions of the principal component analysis. The principal components 4 up to 9 show differences among the samples of Gen01, but Miyagi and Mombasa still appear clustered together. It is only in the 10th principal component, which explained less than 1% of the variation in the original dataset, that we start to see differences among Miyagi and Mombasa samples. The scree plot gives us a better sense of this. Most of the differences in this dataset were observed among cultivars, followed by differences among Genoa 1 samples, and finally among Miyagi and Mombasa samples. This study showed that Miyagi is in fact a mutant of Mombasa. Also, the origin of Miyagi reflects events typically associated with the domestication of agricultural crops, which have never been directly observed and documented in other species. Miyagi gives us a rare opportunity to glimpse into the history of agriculture and evolution. By following Miyagi's trajectory, we notice that science is not a pure description of phenomena as they appear in nature. Rather, it depends on context. Not only are relevant the references and instruments employed, developed in previous research, but also factors such as collaborations among researchers, past events, political dynamics, and interactions with society. Although science relies on it, much of the historical memory of research centers is hardly published. Much of the recollection of this unrecorded history depends on the dialogue among researchers, farmers, and society in general. We need to improve records and documentation in order to be able to build narratives that support our work. This way, we will be able to communicate better. To do that, we should be less concerned with advancements and more with recollecting the events that led to where we are today. This is the only way for us to be able to build a more well-founded, stronger future. This video would not be possible without funding from CAPES and the work of people from ESALC, USP, the Genetics Department, the Statistical Genetics Lab, and Embrapa.